Hello, everyone. Good afternoon. My name is Yara Rodrigues. I'm working at the Sea Institute of Cabo Verde, is the Instituto do Mar of Cabo Verde, where I also serve as a member of the executive board. Today, I will be your moderator for this new session of our Marine Biodiversity Networking Friday from Air Center, which is a very interesting initiative, and we are all very excited to see the session from today. I would like to very warmly welcome you all. It's indeed a real pleasure to have you here, even if it is online. And it's always encouraging to have this kind of sessions to bring us together for meaningful scientific exchange and dialogue within our community. So before we start, there's a brief note. If you have any question during the presentation, please feel free to add them into the chat box, and they will be addressed during the Q&A session at the end. So let's go for the introduction of our speaker, which is my very real pleasure to present Dr. Teresa Amaro. She's a researcher at the Department of Biology at the University of Aveiro in Portugal. Teresa holds a PhD from the Wageningen University. I hope I didn't say it wrongly. And she completed in 2005 her studies. And since then, she has built a very extensive international career in deep sea ecology. And she's also a specialist in trophic ecology and ecosystem functioning across submarine canyons, abyssal plains, and seamounts, and in multiple ocean basins, including the Atlantic, Pacific, Mediterranean, and Antarctic. Her research integrates fields of oceanography, in situ experiments, sediment scoring, moored instrumentation, and seafloor imaging to better understand the deep sea, biodiversity, and biochemical processes under changing climate conditions. So as you can see, she's a very she's our expert today. She has a lot to tell us. And I'm very pleased because the topic that she's going to address today, ecological patterns and biodiversity of black corals and gorgonian communities across depth gradients, is in Santo Antão from Cabo Verde, which is one of the areas that she's been working nowadays, and she's leading research activities in the country, particularly around the island that I said, focusing on the mapping and ecological assessment of the specimens of corals. So without further delay, it's my great pleasure to give the floor to Teresa Amaro, and please join me welcoming her in a very online style. The floor is yours, Teresa.
Thank you. Can you see my presentation?
Yes, we can.
Okay. Thank you so much, Yara, for introducing me, and thank you, Air Center, for this invitation. Without further delay, I think I need to go further because I have a lot of slides. So my presentation will be part of the COAST project, which is a project that deals with conservation of marine ecosystems around Santo Antão, Cabo Verde, that started in 2022. Of course, I'm not gonna go into detail or a lot into detail in this project because I do not have time, but if you want to know much more, here you have the QR codes. So have a look, and then whenever, if you have any also questions about the project, please let me know. So why this project? Why in Cabo Verde? So Cabo Verde is a country where blue growth can help to contribute with solutions for high poverty rates, while building on the long tradition of local economical use of marine environment. However, no valuable baseline knowledge on the environmental status of their marine ecosystems is available, which hampers the development of measures ensuring their sustainable use, management, and conservation. So the COAST project aims to provide a very multidisciplinary understanding about the biodiversity and ecosystem functioning, as well as suitable indicators of recovery, so that in the end we can recommend marine conservation measures. So where is this island based? So Santo Antão is an island that is part of the Cabo Verde Archipelago. This is Cabo Verde, this is Canarias, this is Ethiopia. And Santo Antão is the most western island in the Cabo Verde Archipelago. Why Santo Antão? Because Santo Antão is an island where the local communities highly depend on marine resources, also in agriculture. But as outlined in the blue growth chart, there is an urgent need to collect baseline data on its marine ecosystems. So the project structure deals with a 6 work package. I'm not going to go into detail. We are composed by 10 different partners from 5 different countries. And I'm going to touch base in mostly on work package 1, 3, and 4 in this talk. Coast, what were our questions is, how can coast contribute to developing conservation guidelines and priority strategies for the affected ecosystems? And how can we translate all the data that we're going to have into policy interventions to reduce economic and social disparities, both locally, of course, but also across other African coastal countries? So what are the questions? What do we want? So we want to, how can we deepen our knowledge on these marine habitats of Santo Antão? How can science deliver efficient management recommendations? Because this is actually very hard. How can we improve research capacity to collect all these data? And how can we advance innovative technology and vulnerability analysis of marine ecosystems in Santo Antão? For that, and because in the beginning of the project, whenever we wrote a project, we could not really invite any local partners. We decided to really involve the key stakeholders of Santo Antão, but also in Cabo Verde. So we deeply collaborate with the local community, namely the fishers, the NGOs, but also local environmental authorities, university institutions, and local diving centers. Of course, this project would not be possible if the local fishers would not work in collaboration of us. So back in 2022, I went to Santo Antão and I established a local network of fishers that could help us to collect all these data. So I established a local network in Porto Novo, in Tarrafal, in Cruzinha, and in Janela. And I'd like to emphasize that without these guys and without the colleagues, this project would never been achieved. So going to our title, we started I'd like to start about the marine habitats, subtitle. Back in 2022, we had 232 drop cams, and you can see here in green, and we had 28 diving transects between 22 and actually 24, and you can see in orange. And we could characterize at least 260 sites around the island. As you might know, drop cam videos, of course, lead us to a wider extension, is a bit less precise, but in diving, using photo quadrants is a bit more precise, but of course, there is a lower spatial extension. So based on the ADAPT classification for the Canary Islands that had already 19 marine habitats, for Santo Antão, and between 15 and 30 meters, we classify and identify the habitats around the island. So what did we find out? We found out that seaweeds are everywhere in the island, as you can see in red. Also, rhodolites is, everywhere. So seaweeds and rhodolites are the most abundant. We have corals and sponges, also pretty much everywhere in the island. We have some seagrass locations and also some other fauna, namely some garden eels. You can see some garden eels, well, maybe you cannot really see, but they are here in the photo. Yeah, it's quite unique, these garden eels that we found. Now talking about non-biogenic, of course, the habitats were basically rock or sand or a mix. So, talking now about the benthic communities, but in shallower locations. So, in depth lower than, not lower, higher than 30 meters. We saw that the coverage of benthic organisms were in percentage about 70%, whereas the rest was substract, and these benthos organisms belong to 66 taxa. And what can you say about these is that the highest coverage were in locations more into the northwestern side of the island and in the northeastern side of the island. And in terms of the benthic community composition, these benthic communities were dominated by either algae or rhodolist, as you can see in the graph here. Now, in terms of diversity, we know that the higher the Shannon diversity index, the healthier and the less disturbed the habitat is likely to be. And you can see, so here you have the map of Santo Antão, and you start the number, the sampling sites start here in the north from one, and then go around the island until the Cruzinha, more or less Ponta do Sol site, and you can see that is more or less in the north or northwest that the higher that the Shannon diversity is the highest. So what about the fish? So we did exactly the same, and this is a graph of Santo Antão where you have the total fish counts. We counted more than 41,000 fish belonging to 54 taxa. And as more or less in the benthos, what happens in the benthos, you have the highest counts in the northeast and in the southern western side of the island. And this is a photo where you can see a high count and here is a low. So we did exactly the same. We look at the composition, and you can see that the ichthyofauna composition was mostly dominated by Azurin and Chromus lobocki, that is in yellow and in purple, which is actually very interesting. If we, and also between more or less the same sites where you had the highest coverage of benthos, also here in the fish, you have it, the highest ichthyofauna community happens more or less in the southern east and in the, southern west. Looking at the diversity and with the same, we also had the same partner, and it's also here between the site 16 all the way to 20 where you have the highest. So between here in the Tarrafal and Monte Trigueira, you have the highest Shannon diversity. Now going to actually my favorite site, which is the mesophotic, meaning where we actually found quite interesting patterns here. We found in Santo Antão several sites composed by black coral forests and gorgonians. Here you have a little video, a little example of what a black coral forest looks like. It's very, very interesting. There are many corals, black corals, and it's a site that starts at 37 and goes all the way to 55, this site. It's a very, very interesting, and we're going to talk about it. And this is an example of gorgonian fields that we also found in Santo Antão. It's a place, this is a place located more into the north, and as you can see in the image, it's full of gorgonians, and it was very, very special. So, what did we find is in these black coral and gorgonians? So what did we do? We found 11 species of black corals and gorgonians. Here in the picture, of course, it's only 7, but it's just some examples. Stichopates, Antipatel, and Tanisetipates were actually the most prominent black corals in these black coral forests. Leptogornigaini and Capverdiensis Orionicella granulata and Papilifera were also present everywhere. So now talking about the densities, so I can let you know the densities of Stichopates and Tanacetipates are among the highest reported globally when we looked under literature. The island is characterized by diverse coral assemblages with distinct patterns observed across different locations, as you can see in the figure. In the northeast, southeast, and southwest, black coral forests are proeminent, while gorgonians assemblages are found in the northwest, northeast, and south of the island. Here in this graph, you see that Gates of Aden, which is located here, was the site where we had the highest colonies per square meter. And here you have a picture of how these look like. So for you to have an impression how these look like in the field. Kanjana, which is now here, had the lowest density, and here it's a photo how the locations look like. And Gorgonian Valley had intermediate densities, and it's located here in the northwestern part of the island, and this is an impression of the place. So we characterize the corals, and so this is a graph of a distribution of coral heights across the 14 locations sampled, and this will emphasize the variability and distinct partners among gorgonians here and black coral species. Stichopates, for instance, Stichopates varied, for instance, from a minimum average of 20, more or less 20 centimeters, to a maximum average of 51 centimeters. And also for the gorgonians, the average height of gorgonians across the islands also revealed differences among them. For instance, the gorgonians Capverdiensis, you can see here, had relatively smaller average heights, and these contrasts with larger Papilifera and Gaini, you can see in the lower part of the graph. If now we also conducted trait-based analysis so that we could characterize the functional structure of the studied corals assemblage in terms of their capacity to provide like a three-dimensional habitats. And these had an objective so that in the end we could provide or select sites for, let's say, marine protected areas. So for these, we selected here in this graph, we select 3 functional relevant traits: the growth form, the height, and the skeleton type. And for the coral growth forms, we classified based on their architecture, if it's a branch planar, bushy, or if it's unbranched erect. And these, of course, deals with the influence of the water circulation, food trapping, and habitat provision. So it was because of this as we select these traits. So if we now look into this side of the figure, The trait structure of the examined coral fauna presented 95% of the variation explained within these two dimensions, with the first axis explaining 70% of the variance. So species were mostly separated, so depending on their type of skeleton and morphology, with traits typical from gorgonians, here in this side of the graph. Well, typical like branch planar morphology and presence of sclerotides. And for the black corals, which is on the right side of the graph, are the traits typical from these, with the presence of chitinous skeletons. In the upper part, you can see they were mostly distributed for smaller sizes, like Cabo Verdeensis, Torres corals with larger size and bushy morphologies. So if we plot our sites here in this graph, we mostly, we will have a representation of the functional identity of the different explored sites, which can reveal differences in the trait dominance. So sites from 1 to 9 are dominated, so here are dominated by bushy and unbranched erect black corals with great height. And in contrast, only 2 sites, the 10 and 11 here, were dominated by small gorgonians with branch planar morphology. And sites like 13 and 14 were also dominated by gorgonians, but in this case, these tend to be large, either planar or bushy morphology. So we decide to cluster these. So this is a cluster 1, this is clusters 2, 3, and 4. Then we also calculate a functional richness, so the freaks, so to further understand the functional implications of these traits. So we can see here that places like in green, they had the highest functional richness. For instance, Hanging Garden, it looks like this. And places like in Cruzinha had a very small or low value of functional richness and looks like this. So you can really see the difference. And here are all the sites that we have sampled. So we wanted to do this to select priority sites for conservation. And so how, we would do this? So we decide to protect trait diversity in terms of habitat provision capacity. We decide to at least to select 1 site per cluster, then the highest freak and the highest density. So if we translate these into the clusters, so Gates of Aden or Hanging Garden had these. Also Gorgonian Valley 2, Sphinx, and Cruzinha. So we select 1 or 2 sites in each cluster. So for priority, if somebody would ask me where to protect based on this data, then I would select these places. So the Hanging Garden, the Ponta de Tubarão, and then, the Sphinx, and then Gorgonian Valley and Canjana. But bear this in mind. Within these black coral forests, we also decide to characterize the benthic communities. So within these places, we counted more than 6,000 organisms belonging to 42 taxa. And the most interesting thing was that the highest densities of these communities were dominated by endemic species, the Leptogornia capeverdiensis and the Atlantia capeverdiana, or also rhodolites. So we did exactly the same as we did for the shallower benthic communities. So in this graph, we have the taxa distribution per sampling site. Here you have the graph. And this is the abundance percentage. And what we see here, it's in places located in Janela, Ponta Tubarão, and Hanging Garden, which are here, are more diverse, and also the abundance percentage is highest, where places like in Ponta do Atu, Morro Branco, and Monte Trigo, you have the less, let's say. And so we could say that Janela and Hanging Garden were the sites where you had more species, and these species were in a highest, let's say, percentage. Also Ponta de Peça, which is located here, but the percentage was a bit lower, and Monte Trigo, which is here, it's the site where you had the less. Now, sorry. What did we do after? We wanted to know if there is a significant difference in terms of benthic communities composition among the sites, and we saw that there was a clear difference. There was a significant difference between the east here and the west, which was very interesting. So now we did the same for the fish. We counted more than 26,000 fishes belong to 58 taxa. And also here there was an endemic dominance. And in our video transects, we counted few seriola and sharks. But yeah, it doesn't mean actually that they are not there. So in terms of the community, we did exactly the same. And also here, sites like Janela, Ponta do Tubarão, and Hanging Garden had the highest diversity, but also the highest abundance percentage of these taxa. And in contrast, Ponta do Atum, Morro Branco, Monte Trigo had the less. Janela was the location, so to remind you, here is Janela, was the place where you had the highest taxa diversity, and Morro Branco was the one that had the less. We did the same, and although here in this graph, you can see a complete dissimilarity between the two regions, there was not a significant value. And this could be because of the small sample size, but nevertheless, you can see, yeah, in space that these two locations are different. So we wanted to know more about these species composition. We wanted to know how different our sites are, because these have implications for conservation management. So we calculated the beta diversity for the benthos and for the fish, and the total benthic beta diversity was 2.29. It means a high turnover of species, so the communities are very different from site to site. And because you might know that low beta diversity means that communities look similar across sites. So for benthos, we had a value of 229, and for the fish, we had a bit lower, but still high, which also means that is represents a moderate to high turnover. But if we break down the regions, so if we separate them between the east and the west, we have here higher, so in Tarrafal, Montrigo, and Morro Branco, the benthic beta diversity is higher than in the east, and for the fish happens the same, which is quite interesting. So both benthic and fish communities show a high beta diversity in the south-southwest region, indicating a greater spatial heterogeneity. And this reflects a localized turnover and emphasize the importance of preserving multiple sites across regions. And this was actually very interesting. So if we put this into a map of Santo Antão, and if we put all the data together, so these are the corals, and the corals, these places that are here, were the ones that we would select for conservation. If we put the other benthos and fish, we would also add these sites. So what did we do? We organized several meetings with the stakeholders. We put together NGOs, the fishers, the local authorities, and we presented our data. And we wanted to discuss with all the locals what we should do, where to preserve. So based on a very long discussion and also based on talks from Tommy Mello from Biosfera and Silvana from Terrimar, and mostly important from the fishers, actually, we decided to select a place, is Tarrafal Monte Trigo area, let's say. It's this place for the fishers and the local authorities, a place that we should actually select to be conserved. So, but of course you may ask, but the biodiversity and density is lower. Well, it's still high, but lower compared to the other sites. But the biodiversity is higher. Locals here, they really want to restore this place due to the high densities and diversity in the past. And the ecotourism here in this place, also because of some actions of the local NGOs, the ecotourism is growing. So we believe if we protected this area without any anthropogenic influence and with some ecosystem restoration, maybe the ecosystem will be possibly restored. So yeah. Now I wanted to lead you a little bit to restoration and why we believe that restoration here in Santo Antão can actually work. So we had a pilot case study. We characterized the upper mesophotic reefs dominated by habitat-forming gorgonians and blake corals to select the most adequate species and populations to be used as donors in the pilot restoration action. So we select species like Stichopatis, Tanacetipatis, and Unicella for restoration action. So we had several, we initiate these in October 2023. Here you can see some images of us working in the water, selecting and starting this restoration action with these species. We went back in October 2024 to measure and to evaluate how this was actually working. So for gorgonians, like Unicella papilifera, showed a survival probability of 84%, as you can see here, which is higher than those reported for the most species in previous studies. And for blake corals, like Stichopatis, they showed a survival of 92%, similar to values reported elsewhere. However, Tanacetipatis showed a lower probability, especially when considering that all of those 50% of alive colonies were only partially alive because there was some necrosis, as you can see here in this figure. So for this pilot study, we would say that Stichopatis and Unicella papilifera were viable species to conduct this recovery, let's say. But Tanacetypatis was not a viable species. So we went back in October 24 and we repeat this pilot action for Tanacetypatis, but in a wall, as you can see here in this picture. And what happens is that actually, in October 2025, we did the same as in October 2024. We had a look if the species grew, if they were dead, and actually the values were quite, in the same as in October 2024. So for Papilifera and Stichopatis, they grew and they were all alive. And for Tanacetypatis, actually, they survived and they grew. So it was actually really, really good news. So given the results obtained, so our pilot restoration action worked. And the next step will be to scale up. And maybe as this place was based in the southeastern part of the region, we would like to do exactly the same action, but in Tarrafal, Morro Branco, and Monte Trigo area to see if we obtain the same results. So, if you would like to see more and go into detail, you can have a look at these papers that are already published. However, in our COAST web page, we have many more papers that we already published, and we have several others under review. So, but you can also, email us for any other information. I think I talk too fast. And so I'd like to acknowledge everyone. Thank you everyone for being here. You can have many more information here. You just have to scan our QR code. All the information is in our COAST webpage, but also we had the COAST in Facebook and in Twitter and LinkedIn, but also in Instagram. And thank you. Any questions?
So thank you very much, Teresa, for your very insightful and inspiring presentation. It's very nice to see studies about my country, and I'm pretty sure other people from Cabo Verde that are also here and listening, they feel the same, and very relevant discovered that you found. So now we are open for questions. I don't see here in the Q&A box any question. So it is time now. So I'm pretty sure there's a lot of comments and impressions or small curiosities that you have regarding to the study. So someone is saying that they put two questions, but I cannot see them.
It's in the Q&A.
Okay, so
It's
yeah,
from
okay, I found it. There's a question here from Ana Faria. First, she said it was a very informative presentation. Congratulations to Coast Project and to you. And if you know if the fishing pressure is lower in sites with higher density of gorgonian and black corals, and also if there is any correlation with fishing pressure and biodiversity.
Yeah. Thank you, Anna, for the question. It's a very interesting question, but I have to say that is the opposite. Where we have the highest density and diversity is actually where the highest fishing pressure is. So the fishers, and now I'm talking only about the artisanal fishers, because they only go yeah, they are to those depths, because the other semi-industrial vessels, they go elsewhere. But it's exactly in the places where the highest density and diversity is. So yeah,
Yeah.
unfortunately.
Thank you, Teresa. There are two questions from Mark Costello. What is the annual variation in sea temperature with depth? And if this is considered subtropical?
Yeah,
And another question is if you have published the species present into OBs
yeah,
or GB, especially in endemic species.
yeah. Thank you so much, Mark, for the questions. Starting by the variation in temperature, yes, there is an annual variation. So from January, you have temperatures about 20, 21 degrees, and then it starts increasing up to June, July, up to October or November, where you can have temperatures of 26, 27 degrees, and then it starts decreasing again. And this has to do with the Canaria current influence. And in depth, on the surface, you have more or less the same temperature, 20, 21 in January. Also, you go below, and it decreases more or less about 1 degree Celsius. So yeah, this is the annual temperature. And about the species presence, so my PhD student and I, and also Jessica, we actually did a catalog of species, and this is already published in OBIS, and yeah, and it's all there. So it's a catalog with all our results. And also Ariana from Barcelona, she's putting all the data from coast, not only in terms of species, but in terms of the temperature, oceanography, also yeah, social and economic. Everything is in OBIS. So yes, it is.
Thank you, Teresa. There's another question asking why would you, how did you find the black coral surviving in the second approach? Why would you say that
Yeah.
this happened? So how would you
Yeah.
justify that?
It's because we have measured. I do not have also more pictures of this, because this is a very recent work, and we measure all the Thanasatipates, and there was a difference. So they grew, and there was no necrosis whenever we observed the same species restored. So that's how we evaluated. They survived, and they grew, so they were good for restoration.
But then, how do you justify the necrosis in the first step? So do you have any idea?
Yeah, you can see visible that they were dying. I can
No,
share.
why? Why?
Why they are dying?
Yes.
We actually in the beginning we put Thanasetipatis on a very flat environment, and although you can see in Santo Antão some Thanasetipatis, in a flat area, the highest densities and where they appear, they are mostly on a wall. So I think it is related with the currents and with the food. So they survive better on a wall than if it's flat. And perhaps it also has to do with the light, because if it's flat, maybe you have a higher light intensity than if you have the black corals on a wall. So I think it's for this reason.
Okay. I will check if there are other questions. I will pay attention to the box. But in meanwhile, there are some small curiosity also
Yeah.
that I have apart from that. So you were showing these differences between the gorgonian and the black coral area and patches. Um, did you measure apart from the survival the rate of growth?
Yes, yes,
Okay, I
I did.
didn't pay
But
attention in that.
yes, yes, yeah, and I did, and I put it in one of the slides.
Okay.
But I can. We are already preparing the paper, but I can share with you the results of that growth rate.
Yeah, actually, I'm very interested to know if the growth is higher or it's medium, like is expected for the species that you
Yeah,
had in the study. Okay.
yeah, but if you have a look, sorry, I'm gonna so here, there. Here, so the stichopathies, you have the mortality. Here, this is the survival. But if you go here, yeah, actually I didn't put it, sorry. Yeah, we had a graph, and I decided not to put here because I already had, sorry, yeah, I already had 55
That's fine.
slides, but I can share that with you.
And combining all the parameters that you did in the study during all of these years, how do you say that those differences that you find in between the two habitats of gorgonians and black corals, in what way does it contrast with the spatial distribution for the habitat preference, and how the parameters can influence that also?
Sorry, what do you mean?
So having in account the gorgonians and the black corals,
Yeah.
you found them in different habitats, right?
Yeah, and I found
They
places
have
where they were
patches
all
where they
mixed.
combine.
Yeah,
Exactly.
yeah,
So how can we perceive this spatial distribution, but also within the framework of other parameters, oceanographic parameters that can be considered?
yeah. If you have the map of Santo Antão, sorry, in mind, let me see. Let's share again. I think, let's see. Well, you can have this map anyway. Wait, uh, sorry. These are the habitats. I'm sorry, I'm trying to look at the map of the corals. So you have here, this is the map. And so this is places in blue where you have the highest densities of black corals. Here you have highest densities of gorgonians. And in all the places you have a mixture, although here in the cruising area, let's say, you do not have black corals, and I cannot explain why. But what I can tell you is that the current and the wind influences quite a lot in this direction. So from Ponta do Sol all the way to Janela, and it's very cold, and the waves are and the currents are very high. Also here, but then only from 11 o'clock onwards. But the influence from the currents, it's not as much as you have it here in the north. Here, as you know, in Tarrafal and in Monte Trigo area, rarely you have the influence of these currents that comes from the north. So it's only when you have the currents that come from the south, and actually these currents, they do not happen that much. It happens mostly in between January and March. And here, which is very interesting, number 5, number 3, you have the influence of all the currents. And I think that's why you have a very high diversity, but you do not have so much densities of the corals. Then you have here in the gorgonians. This is an area that is very hard to sample because, as you know, the sea is actually very rough. You have very high currents, you have really high winds, and you have very high waves. So it's quite hard to do anything in this area just in some months. And I believe that these gorgonians that we found here, they are really happy there because you have lots of food, lots of lots of food, and I think that's why they are there. But why you do not have black corals here? And I think it's because up to 30 meters more or less, Yara, I did not find any walls. And the continental platform in that area is very long. And so it was not possible because I only used the artisanal fishing boats, and it was impossible for us to go with the rebreather diving, to look for a wall or for a drop off. But I'm dead sure because I was there
Okay.
with Geomar to study the deep sea, and we also found black corals everywhere and gorgonians, of course. So I believe that they are distributed more, on walls than on the flat. So yeah, it's all related, and it's quite interesting.
No, thank you. It's a very good thought on that. So you guys basically had this approach of going for Monte Trigo because of all the characteristics and then bringing forward the restoration concepts. But also, do you have any idea of what could be the next steps in Janela, since there's these very good results with the richness and the diversity
Yeah.
taxa test?
The problem, Yara, is that in Janela there is a very high fishing pressure. It's impossible because these, although I'm new in marine protective areas, I think Mark is there, he's a specialist, I'm not. But this has to be done together with the fishers. And if they do not accept, this won't happen. And yes, if you ask me, I would protect those places that I mentioned, Janela, Hanging Garden, and Ponta do Tubarão, that they are amazing. But it's where the fishers are, and they will never agree to protect those areas. So, but Montrigo and Tarrafal, it's the opposite, So the fishers, they really say that, in the past we really had high densities, high diversity, and fishers from Tarrafal and Montrigo, now they have to go somewhere else. And as you know, they go to the seamounts, whenever it's possible.
Yeah.
So they are really interested in protected an area like that because they wanted to have the ecosystem restored. Yeah.
Totally agree. I was very interesting to know
Yeah,
to
but
know what would be your thoughts on that. As you
yeah,
know,
but
we do have also ongoing studies regarding to that also here. Yeah, I'm very happy with your presentation, with all of this work that is being done in the country and in this area. Also to say that we are going to do a partnership very soon. So very soon we're going to keep on the conversation. If you guys have any other question, I think this is the last minutes that we have. Marco is here saying how lovely the videos and photos are.
thank you.
And good ways to prioritize which areas to protect. Okay. Okay. So maybe just one more very small question, Teresa. You said something about the drop cams that you use. It was a lot of use of drop cams during all the region.
No.
Um,
Yeah.
can you give an extra comment on that and how you want to make this more robust in the next levels of the project?
Yeah, so we had a very small drop cam to classify the habitats, but I also felt the need to involve the locals and the local capacity. And so what I did was to write with other partners a project in England with English money to actually improve your capacity building. So we had built a very large drop characterize, and I didn't present these results here, everything up to 100 meters. So we did more than 200 transects between Tarrafal and Montrigo, and we are still analyzing this data. And this drop cam, Yara, is in Cabo Verde possession. So the objective of this project, it's called BAIT, was to improve your capacity building. So it was donated to Biosfera, but because they were a partner and because they really help us to do this, and for this drop cam, we use the vessel from Biosfera. For a very small drop cam, Yara, we use a local fishing vessel. And then we imported all of that in Beagle, and then we just analyze the images like we analyze for the other parameters. So, but if you want to use the drop cam, Yara, it's in possession of Biosfera, but it's for everyone in Cabo Verde.
So you would still use the drop cams for the next steps of the projects if you have so.
Yes, but you have, and you might ask, or somebody might ask, why not an ROV? But as you know, and as you could see, you have lots of walls and you really have very high currents. So I would use ROVs and drop cams, but with very careful because they could get entangled in these walls. So you really have to know and to have the feeling where these walls are to use the drop cams, otherwise you will lose it. You lose the material. So yeah, with caution, and I would recommend use all the methods available. Drop cams, not the small ROV. I would use the ROV for deep sea work, and use diving like we did.
So thank you very much, Teresa, for everything, for presentation and for answering all of these questions. Also, thank you very much to all of you that are here for all of your questions, your contributions. And I think I would like to conclude this Marine Biodiversity Networking Friday session. It's a very nice initiative. Again, you should continue, and we would like to look forward to welcoming to the next session. So have a great day, everyone, and thank you very much.
Yeah, thank you. Bye.
Ciao, ciao.
Mm.